And my name is Priscilla Ramirez. Today I'm going to be talking about high throughput screening for drug discovery. This is my local expert module 5 video submission. So we briefly talked about high throughput screens and how they're used. We talked about this in module 5. And one of the slides in module 5 said, what can you do with cultured cells? And we determined that if you infect those cells, you can use high throughput screens as an analysis method. And you can also, if you mutate the cultured cells, you can also use high throughput screens in order to do this type of analysis. So I just want to talk about the importance and how HTS being important can lead to drug discoveries and medical discoveries for this type of research. So what is high throughput screening HTS? It is a method that can test many samples at once and provide a high rate of approximately a few thousand compounds per day or week, and even potentially millions of samples per day or week, or millions of compounds per day or week. So we talked about something else that was high throughput, and that was, I believe, in module 3 or module 4, when we talked about Illumina sequencing, and that was one method of analysis that was considered a high throughput sequencing method. So why is HTS important, or why do we want to know about it? And it's to better view the biological activity within a pathway at the cellular or molecular level and within an organism. So if we can understand how, and we're going to talk about this with drug discovery, but if we can understand how drugs affect molecular or pathways in the body, and if we can see how drugs impact the cellular or molecular level or the behavior of cells, then HTS can help us to determine that. It can be a way of analysis in order for us to see the effect that certain drugs have on cells. And so cell lines are frequently used during high throughput screening. Why is HTS important in drug discovery? So HTS is important in drug discovery and is a method, it's a popular method and a leading method because it determines the phenotypic response to drugs. So you can do 2 different types of discoveries or base tests. You can do the target base or the phenotype base. And we're going to talk a little bit more in the next slide about phenotype based drug discovery. But essentially, for the phenotype based drug discovery, you're going to do an assay. You're going to develop your assay, and then you're going to screen your cells. And then you'll do the target deconvolution. And then from that step down are the same steps that you would do in a target-based drug discovery. So in a target-based drug discovery, you select for a certain target, and then you perform the development of your assay. And then rather than cell-based screening, there's going to be biochemical screening. So pretty much we would be a little more, since we're tying this in with what we learned in module 5, we would be more focused on phenotype-based drug discovery because of the cell line and the cultured cells that are being used. So you can also determine drug activity or anticipated drug activity in the cell, like anticipated drug behavior, or whether a drug, like we mentioned previously, whether a drug reacts differently in a certain pathway. And you can also characterize the metabolic and pharmacokinetic and toxicological data about new drugs. So actually, this picture on the right, I got it from a toxicological I got it from a toxicology, that's the word. I apologize. I knew that wasn't the word. Toxicology research paper. And so they were determining the different characteristics that they see when they do perform HTS. Again, I apologize for the hiccup. So this picture on the right is from that research paper, and it's a typical high content screening workflow. So these experiments are designed and cells are plated accordingly using the liquid dispensers into either a 90, so using a pipette into a 96, 384, or a 1536 well plate. So if you're putting your cells into a 1536 well plate, you better believe you're going to get, if HTS is successful, you better believe you're going to get a high throughput. So we'll talk a little bit about the screening goals. So the screening goals, like we talked about, you can do target-based and phenotype cell-based research, and that can lead, the main goal of that is leading to medicine development. And in this figure on the right, excuse me, it is a possible decision diagram for selecting an HTS technology. And that highlights some of the major considerations in the process. So cell-based screens, focusing more on that, cell-based or organism-based screens, they're used when, one, there's a desired molecular target that's not known. Two, the target can't be adequately reconstituted in a biochemical assay. And three, the desired phenotype only exists in a cellular context, such as differentiation from one cell type to another. So in the second explanation, the target cannot be adequately reconstituted in a biochemical assay. So that already rules out going under the target-based drug discovery. We wouldn't be able to do that because the target-based workflow said that you have to create that biochemical assay. So this is just showing a little bit about the cell-based assay, and the cell base is on the bottom form for the phenotype cell base. And so you can see on this bottom portion of this figure, on the right-hand side, if it does lead to cell death, you would want to go through this diagram. And on the left-hand side, if it doesn't lead to cell death, you would want to follow the steps. And on the top is if you were doing the target-based drug discovery. So cell-based, you can do multiple targets screened at once. And we already went through a little bit about why, or when and why they are used. If you have any questions, go ahead and drop them in the comments, but that is all that I have for now. Thank you for watching.